My name is Barbara Melchert and I'm from the Department of Molecular Pharmacology of the University of Groningen. Today's date is March 30th, 2020, and this is a recorded class for the course Pathology and Immunology for first year pharmacy students. I originally gave this class on March 3rd, but I re-recorded it to have a set of complete online lectures. So this class is about B and T cells, basically about the adaptive immune system. In the other videos I talked about the innate immune system, but if the innate immune system needs more help, they can involve the adaptive immune system. Here shown the phases of immune cell infiltration. So first of all you get the neutrophils that will help out with an infection. Then it becomes a bit messy, you need to clean up stuff, you get monocytes infiltrating, but if they cannot contain a threat, the help of T cells is needed, and then you get infiltration of T cells. So the neutrophils come in the first 6 hours, then within 24 hours you get monocytes, and it takes about 3 days to get your T cells in. And your T cells are a form of very specialized help that can really specifically find and kill certain threats. And there are two parts of the adaptive immune system. Basically there's humoral immunity consisting of B cells and cell-mediated immunity consisting of T cells. So the humoral immunity is directed against extracellular microbes. It's B lymphocyte making secreted antibodies that can block infections and eliminate extracellular microbes. And cell-mediated immunity is consisting of T cells, either T helper cells or CD4 positive T cells, or cytotoxic T lymphocytes, or CD8 positive T cells. The CD8 positive T cells can directly kill cells that are aberrant, so infected cells or tumorous cells. And T helper cells, CD4 positive T cells, can help other cells in killing infections. So for instance, macrophages that have eaten microbes. So how do B cells recognize antigens? Well, they do this through the Bcell receptor. And the Bcell receptor actually is an antibody that's membrane bound. So these membrane-bound antibodies can directly recognize all sorts of antigens. They can recognize proteins, carbohydrates, lipids, many types of foreign antigens. And since the membrane-bound antibody is on the outside of a cell, the response is always directed against an extracellular antigen. T cells have a slightly different way of recognizing antigen. They recognize through the T cell receptor, which is not an antibody. And T cells can only indirectly recognize antigens. And these antigens consist only of proteins, and in particular peptides, so parts of proteins. And T cells can only recognize these peptides when they're presented to them. So they need antigen presentation. And who does antigen presentation? That's other cells. Other cells can present antigen to T lymphocyte in the context of certain molecules. So therefore the response of T cells is always directed against intracellular antigens. Here the structure of the B cell receptor, as you can see, an antibody which has a Y shape is bound to the cell surface of a B cell. So a B lymphocyte can recognize 2 antigens per B cell receptor, because an antibody always has 2 antigen recognition sites. Either arm of the Y can recognize an antigen. And that's shown in more detail here. This is what an antibody looks like. It's 4 proteins bound together, 2 smaller chains called the light chains and 2 bigger chains called the heavy chains. Those are put together in a Y shape, and the part of the heavy chain and the light chain on the outside is where antigens can bind. This is the antigen recognition site. This tail of the antibody is called the Fc region and is what binds to cells or to Fc receptors. And this part of the antibody, which actually recognizes the antigen, is called the Fab region. And here you can see the antigen binding site, that it can bind to the antigen, and that it can actually bind to 2 antigens because there are 2 antigen binding sites. Now, antigens that are being recognized by this B-cell receptor can activate the B-cell receptor. When this B-cell is activated, then it turns into a plasma cell, and this plasma cell can actually start to produce antibodies that are soluble, not bound to the cell membrane anymore, but are released into blood and tissue fluids. And then there are different types of antibodies a B-cell can make, and those types of antibodies determine the immune response. So what types of immunoglobulins do we have? Well, we have the membrane-bound ones that are always monomeric, so it's always one Y. And a B-cell has, so the B-cell receptor, which is an IgM type of antibody, and it also has another antibody, which is an IgD antibody, which looks like IgM, but is slightly different. So the IgM is the B-cell receptor, and IgD, it's not clear what that's for actually. Of the soluble antibodies that a B-cell can produce, there are 4 types. There's IgG, IgE, IgM, and IgA. So 2 of them are monomeric and the other are multimeric. IgM is a pentamer, so it's 5 molecules together, and IgA is a dimer, it's 2 molecules together. And IgM is actually the stuff that the B-cell receptor is made of, and what the B-cell can produce then in soluble form as a pentameric molecule. B-cells can also produce other types of antibodies after they have gotten help from T-cells. Then they can switch from IgM to other types of antibodies, IgG, IgE, or IgA. So what do these antibodies do? Here we have the B-cell. It recognizes a bacterium, it becomes activated, becomes a plasma cell, starts producing antibodies. And what these antibodies can do, they can neutralize the threat by binding to it. And then this can be recognized by phagocytes and phagocytosed. Or it can activate the complement system, and then the membrane attack complex here will lyse the bacterium. Here is opsonization again, so the bacterium is coated with antibody and can be phagocytosed. And what can also happen when a cell is become aberrant and it's recognized by antibodies, the cell is coated with antibodies, and these antibodies can activate a natural killer cell, an NK cell, and then this natural killer cell will start to kill this aberrant cell, and this process is called antibody-directed cellular cytotoxicity, also known as ADCC. What can the different types of immunoglobulins do? So IgG is the most versatile antibody. because it can opsonize, it can activate complement, it can neutralize, and it can induce ADCC. And the other ones are a bit more limited. IgE can only activate mast cells and eosinophils. IgM can activate the complement and can neutralize threats. And IgA is mainly there for neutralization and is only present in the mucosa, is mostly present in the mucosa, and is therefore important for mucosal defenses. So how do T cells recognize antigen? Well, that's through the T cell receptor, like I said, and they can only recognize this antigen when it's presented to them. This can be done by normal cells, but there are also very important antigen presenting cells, and I already talked about those. Dendritic cells can present antigen to T cells, and then the response is directed against intracellular antigens. So here's the structure of the T cell receptor. It is slightly different from an antibody. It only has one recognition site. And here the two of them are together. So here's the T cell receptor with one antigen recognition site. And here's the B cell receptor with the two antigen recognition sites. So they're quite different, but there's one commonality. The number of specificities is almost limitless. That means that every lymphocyte is monospecific. It means it can recognize one particular antigen that another lymphocyte cannot recognize. So if we have two blue cells here, for instance, then this can recognize a bacterium, and this one can recognize a certain molecule that is expressed by fungi. And then if this one recognizes the bacterium, these four receptors all will recognize that same bacterium. And if this one that is not here, but imagine it being here, recognizes a fungi, then all those receptors will recognize that fungi. And so we have billions of T cells that all recognize something else, and we have billions of B lymphocytes that all recognize something else, something very specifically present in molecules. Here again, what antigen presentation is. Here we have a T cell with T cell receptors. It cannot directly recognize the antigen. You won't get a T cell response. It only recognizes the antigen when there is a cell that is presenting the antigen to the T cell. And this antigen presenting molecule is called the major histocompatibility complex, or MHC molecule. And in humans we also call these molecules HLA, human leukocyte antigen molecules. These molecules are used to present part of a protein to the T cell. And if the T cell then recognizes this presented peptide, then it can become activated, and then a response can start. So this major histocompatibility complex, or in the human leukocyte antigens, basically is like a presenting tray for T cells. They want their food presented to them on a tray, otherwise they do not respond. So why are these molecules called major histocompatibility complex? Because for the longest time we knew that these molecules existed, but we didn't know what they were for. It was just noticed that in organ transplantation they made life difficult, because if people had very different types of these molecules, then the organ transplantation wouldn't work. And if their molecules were very similar, were compatible, then the transplantation would work. So that's why they're called major histocompatibility, because if the tissues were compatible, if these molecules were similar, then a transplantation would work. And now we know that they're actually there for antigen presentation. But at the time of discovery, they were linked to organ transplantation. So what do these molecules look like? Well, actually we have two types. We have major histocompatibility complex 1 and 2. And they're slightly different, and their expression patterns are also different. MHC class 1 is present on all cells except red blood cells, because red blood cells don't have nuclei, so they cannot express MHC class 1. But all other cells have MHC class 1, and it looks like this from the side. It's a bigger peptide with the alpha peptide with a smaller beta peptide. And if you take a top view, then it looks like this. It sort of looks like a tray, and this purple thingy is the peptide that can be presented in this tray. So the pink part is the actual MHC molecule, and within that molecule is a peptide that is shown to the outside world. MHC class 2 is only present on antigen presenting cells or phagocytosing cells like dendritic cells, monocytes and macrophages. It's slightly different because the alpha and the beta chain are similar sized, and if you take the top view, both the alpha and the beta chain are part of the tray and present the peptide. So MHC class 1 presents slightly smaller peptides of 8 to 9 amino acids, while MHC class 2 can present bigger peptides of 10 to 30 amino acids. Why do you have these two molecules? That's because they express, they present antigens to different types of T lymphocytes. Remember that I told you there are two types of lymphocytes. There are lymphocytes that tell others what to do, CD4 positive T cells, and there are lymphocytes that can kill, the CD8 positive T lymphocytes. So the CD4 positive T cells can help macrophages, CD8 positive T cells, and actually B cells as well in their functions. And CD8 positive T cells kill infected or tumor cells. And MHC class 1 and 2 direct these two types of T cells. So MHC class 1 can communicate with CD8 positive T cells. So since MHC class 1 is present on all types of cells in the bodies, all cells in the body can tell the CD8 positive T cells that they need to be killed, because CD8 positive T cells kill other cells. So if a cell is infected or it's changed and it's not functional anymore, it can present peptides to T cells on MHC class 1, and that's a signal for the CD8 positive T cells that they need to kill this cell. This cell here can tell the outside world, or the CD8 positive T cells, that I need to be killed. I'm a danger to this body. MHC class 2 is only present on antigen presenting cells and can communicate with CD4 positive T cells. And CD4 positive T cells help other cells with killing. So if a macrophage has eaten bacteria and it needs some help killing these bacteria, it can tell through MHC class 2 the T cell that, hey, you know what, I need some help killing whatever I've eaten, so help me out here. So it doesn't want to be killed itself because it's doing its function, but it just needs a little bit more help. So with MHC class 2, phagocytosing cells can tell T cells that they need some help with killing whatever they've eaten. And here it's shown in a different way. So this is a CD4 positive T cell that is responding to an antigen presenting cell that is presenting antigen in MHC class 2. It needs some help killing whatever it has eaten, so it tells this T cell to help out. So this T cell will then become activated, start producing cytokines that can then activate this antigen presenting cell and then help out with killing whatever it has eaten. So both macrophages and dendritic cells can activate or can get T cell help after phagocytosis. And dendritic cells do this in the lymph nodes, and macrophages do this in the tissue. So the way it works is that dendritic cells sense the danger in tissues, eat whatever is dangerous, go to the lymph nodes, tell the T cells, hey, we need help. The T cells become activated, then move out of the lymph nodes to the tissues, and then in the tissues the macrophages can tell them again, hey, help me out here. Look what I just ate. And then the T cell can say, hey, here, let me help you kill that thing. So that is for extracellular stuff that is being eaten, but how do you know that there is an intracellular problem? Because here we have normal cells, nothing wrong with them, but if the DNA is damaged and you get an aberrant cell that grows a tumor, how do you know that this is a problem, right? And that's when you need CD8 positive T cells, because these tumor cells usually grow very fast, and then the proteins that they make are slightly changed. So these proteins are then presented in MHC class 1. So here we have the malignant cell producing slightly different proteins, or a virus infected cell that is producing viral proteins. Then this cell can tell the outside world through MHC class 1, look, I have a problem, you have to kill me. And this CD8 positive T cell can sense this antigen and think, oh, wait a minute, that cell, there's something wrong with it, it needs to be killed. So the major histocompatibility complex, we have MHC class 1, which is present on all cells except red blood cells, and is communicating with CD8 positive T cells. And we have MHC class 2 on antigen presenting cells or phagocytosing cells, and those are dendritic cells, monocytes, macrophages, and also B cells, a special case of antigen presenting cell that we'll talk about more in detail in immunopharmacology, and they can communicate with CD4 positive T cells.